I did that. That's it. That's all it is.
It seems more complicated than it is.
On a computer, yeah, until you
Yeah,
in
I'm-
class.
For me, I'm just trying to like translate it into like, for me, when we're doing this, I'm like, I get it, but then when I get onto a test, it feels like it's like one step above where I understand it.
Yeah, but
Because
one of your
it's
classic
super
questions for this that you've asked multiple times is what reaction can happen in the muscle? And that would be the production of glucose because it goes to pyruvate instead of glucose, right?
Well, the muscle doesn't generate glucose from glycogen.
Only in the, well,
It's glucose 1-phosphate and then it's converted into glucose 6-phosphate. Never takes that into free glucose.
yeah, okay. Got it.
You see that?
Okay.
Does it make sense now?
Of course.
So, and you can see it's still the GP and the DE. You know, the phosphorylase.
And the
And
debranching.
remember that this is just an illustration. If you're gonna write the reactions, the phosphorylase will use PI. That's why we get the G1P because of the PI. However, the debranching enzyme uses water. See that? So if you're gonna write the reaction, there are two different reactions, and you need to be familiarized with the reactions. If you're gonna represent the phosphorylase reaction, is you can put a glycogen. I'm gonna use the same format: glucose N plus PI. Okay, this is the phosphorylase reaction. This is gonna generate glucose 1-phosphate plus glucose polymer with 1 glucose less. You see that? So this is the phosphorylase reaction. How many of these guys we generate? Well, it depends if we want to remove all of them. I put some numbers in there. And the reaction doesn't change whether we have 50,000 glucose residues or whether we have 30. You know, you see what it means? It's always gonna do that. So this is the phosphorylase reaction. How about the debranching enzyme reaction? The debranching enzyme reaction, okay, we're still working on the glycogen particle, okay, but now this is gonna use water, okay, and what's gonna happen is we're gonna generate glucose N-1, and then this is gonna generate free glucose.
That's a bifunctional.
Yeah, because when you look at the process, and now down here I can show you, they know more about this enzyme than their own mothers. I'm talking about the scientists that have done research with this. They've been studying this enzyme for many, many years. And what this enzyme, think about a branch like this. And what this enzyme does, when I use my finger as a dephosphorylase, what it does is do this. Grab this glycogen polymer, you know, in the tears at the ends, and then grab this, okay? But it breaks right here, you see? It grabs like this, but it breaks right here. You see that? So what happens, and then it gets moving. So this is released, and then it moves a little more. But then when it gets in here, it gets stuck. You see that? Gets stuck.
Is that why it stops for prior?
That's what the distance between is. I'm gonna use my hand. It's almost like doing this, you know, and it's grass over here, but it chops right here with the PI. And then eventually it moves along very rapid until right there, because it stops over here because it cannot see any more glucose sticking out to cut with the PI.
That makes sense.
Okay, and then remember that I have one phosphorylase on this tier and I have all the phosphorylase in this tier. Because all those ends, on those ends, I'm going to be binding to the phosphorylase. I'm gonna show you a glycogen particle to make a case about the degradation. I like this one better. Okay, let me see. Let me see, what is the one? Let me put it like this. To look what, to find what.
Is it this one?
No, there is this one.
Bingo.
Because you can see the ends better. Visualize when this is gonna be degraded, the phosphorylase is gonna be attacking or attaching to this. It's gonna do bingo when it's far away. It cannot go any longer. But at the same time, we are degrading this. It's always degrading this and degrading this. That's why the glycogen particle can be degraded very, very rapidly. But then when this phosphorylase gets over here, it cannot continue unless we bring the debranching enzyme. So I'm gonna remind you what the debranching enzyme is. It's a bifunctional enzyme.
Oh, those are all alpha 1,4 linkages.
Yeah, all those, the phosphorylase is can only break the alpha 1,4.
And then when we need to break the alpha 1,6, we need.
And this, look at this is what it does. That's why I think about my fingers like this. You see that? This is the branch. And then, look at this, 4 and 4. Cannot do it anymore. So that's why the debranching enzyme has two activities. There is a transferase. So what it does is take these 3 and put it on the other end and leave just a single one in green alone. But if you have to write a reaction for the transferase activity or the bifunctional enzyme, it will look the same. If you want to write this, the same number of glucose residue will be like this, and it's gonna go like this. Because the number of glucose residue, that will be the debranching enzyme transferase reaction.
Oh, I see.
You see that? When you look at the number of glucose residues between this and this are the same.
You wouldn't write it down because it's the same structure.
Yeah, you look, it's the same, so there is no way that you can, this is a tricky question.
So it looks like a single process, but it's actually two.
Yeah, it's doing, because basically this is represented the number of residues. It's not telling you what proportions, which ones are in one end, no, it doesn't tell you. So the only way that we can show the net reaction of the debranching enzymes by the hydrolysis. And that will be what I wrote over here. This is the net reaction. So the only way that you can see a change is when the hydrolase activity is acting on removing that single glucose molecule. You see that? That's why we need two enzymes. And remember, we don't have a lot of imagination. Chemists, biochemists, okay, how do we call the enzyme that remove branching? Okay, let's call it debranching. Oh, that's so amazingly clever. We don't have imagination, we just call it the way it is, which is good, it's good. The enzyme that makes ATP is called ATP synthase. I think this is brilliant. Why would I make anything, difficult when we can call it the way it is? What else can I show you? Do you want me to show the synthesis?
Yeah, I had a question about the synthesis.
Let me put a clean page. Anybody has a question, anything about this?
Oh, that's good. You drove it home.
Okay. Let me bring a clean page now. So remember, when do I store a little bit of glycogen? When? That's the beginning. When do I store a little bit of glycogen?
In the liver?
No, when, not where.
Oh, when you eat.
Yeah, when I eat what? When I eat
When you eat
lasagna. Okay. So, and you can be, I'm gonna put this as a membrane. You see there are two membranes in here. One is the muscle and one is the liver. See, that's perfect. See that? So here is the lasagna. And lasagna is gonna generate glucose in the blood. Here is the glucose. And this glucose has to be internalized by both the liver, I'm gonna say the liver. No, the muscle is whiter, so I'm gonna put this as muscle. And this is heart. Remember, this is heart and muscle, okay, skeletal muscle, and this is the liver, right? So here it is. It has to be internalized by both cells. So I'm gonna put a protein in here, right? And I'm gonna put another protein in here. This is the carrier, this is the transporter for glucose. So just internalizing the glucose, there are some differences. You have to be ready because you don't know what those guys are gonna ask you, including this guy right here.
Exactly.
So if you were in my office, for instance, how can I help you? Well, I'm concerned because I'm having drawings. Well, don't worry about it. Make sure that you know everything and you're gonna be okay.
Oh, that's messed up.
And of course I laugh, and then the student relaxes a little bit. Okay. So, and that's basically what it is. So we went through this. So what if this is the difference between getting the grade that makes you happy, but this is the one that allows you to be licensed as a practitioner?
It's not about grades, it's about understanding.
Well, it's also about understanding, because that's the other thing too. And how many transporters are out there for glucose? You know that every single cell is gonna have 1 and 3. You should know that. So I made a list of proteins that are found in all cells, and you know that there are no more than 30 proteins. You need to know how many bonds do you need to know.
200.
And you need to know not only the names, you need to know, what they do, where are they located, Well, why you cannot do that with proteins? You make a list of the proteins that are found in all cells, it's just about 30. The ones we discuss only.
Mm-hmm.
And that's all you need. So is that too difficult? Well, I don't think it's too difficult. So when you say, okay, you can name a few proteins that essentially are found in all cells. And then if you go to red blood cells, it's even sweeter because it's less amount. Since there are no mitochondria in red blood cells, then all those mitochondria, there are no enzymes of the Krebs cycle, there is no enzymes of the oxidative phosphorylation and so on and so forth. Mm-hmm. So, but I'm gonna do this. So remember that the liver, mostly when this glucose goes up, this is gonna be done by GLUT4, GLUT2. And this guy is gonna be GLUT4 that is only recruited to the membrane of muscle cells and fat cells when insulin is up. This is the only glucose transporter that depends on insulin.
So water soluble, fat soluble.
Yeah, what do you mean water soluble, liquid soluble?
It's the transporter.
Didn't you say that water soluble goes to the liver via GLUT2, and then fat soluble
Yeah,
goes to the muscle via GLUT4?
What are you saying? What water soluble? I don't
The nutrients. I'm talking about nutrients from eating.
No, all of them eventually get to the liver.
No,
But we're talking a very single nutrient.
you're just talking about glucose right now.
Yeah, we're talking about glucose because we're talking about glycogen synthesis, right? So what happened after glucose? I'm gonna put G's for simplification. Look at this. What happened is converted into glucosic phosphate, and all cells can do this.
He's just talking about glucose right now.
I know what you're trying to say, Steph.
Yeah.
You see that?
But he's just talking about.
Yeah,
This
just
is
like
why I
with the
get confused,
glycogen
'cause I'm like-
synthesis,
I put
because you
everything
know it all,
together.
yeah, but you gotta break it, yeah.
No, there was a study. We should have a single class, a single unit. You see what I mean? We always talk about that unit. So we talk about every single one.
Yeah, we do.
You see what I mean?
Yeah, but he's trying to tie it in because, like you did say, as far as for nutrients and stuff, whatever, we're good.
Yeah.
And so what's the difference? Because there is another difference too in here. Why? This is gonna be mostly done by glucokinase. This
the
is
other
gonna
one's
be done by
hexokinase.
glucokinase.
Right.
Because this is a difference. There is no
Because of the
glucokinase in muscle.
Correct.
That's 4 and that's 1, 2, 3, right?
Correct.
Yeah.
1 and
Okay.
3. Yeah.
And then what happened after that? What happened after that? This is I summarize, and this is only a summarize. When we're gonna do all the thing like glycolysis, because remember this, also these guys are gonna be converted to pyruvate.
That's my problem. I try to put everything together.
Okay? And after this, what we do, we activate, because this glucose cannot be attached a glycogen particle, we have to make it harder. And this is what we do, we activate this. And the activation is the same in both. And now this is this glucose is hot. Now all we have to bingo what we know. Now we're gonna bring the synthase. What the synthase is gonna do, it's gonna take a little bit of glycogen, gonna put it like this, right? And it's gonna add 1 glucose to this glycogen. You see that?
Yeah, that's the N+1. Yep.
Plus 1.
Yeah.
And the same over here. We do this activation, UDP glucose. Okay, and then the synthase is gonna do exactly the same. The synthase is gonna come over here. And it's gonna generate. So basically what the synthase does is add more glucose to a glycogen particle.
So is that the branching enzyme part of it?
Yeah, of course.
Okay.
With UDP.
With UDP.
So that's basically what this is.
Like every time there's a UDP, it adds,
Okay,
right?
Is that true?
the what?
Every time there's a UDP.
Whenever UDP is present, does it continue to add on more like
Yeah, of course.
glucoses? Okay. Okay.
Because how many times, for instance, I can use, how many, for instance, if we're gonna make a typical glycogen particle, how many times is this reaction repeated?
However many times.
Well, let's say it's a typical
depends.
with 50,000. How many times is this reaction catalyzed by the synthase is gonna be, so this is glycogen synthase. And remember that there is a product of this, which is UDP. So the UDP basically is released. You see that? Because that's the reaction. So I'm asking you, for instance, if we will not make a particle that have 50,000 glucoses, how many of these guys do we need? How many times this is gonna be repeated? Remember, this is a game. This synthase can only make the alpha-1 4 linkages.
Doesn't it depend?
I mean, in your PowerPoint, you got 3 arrows. So,
Well, the synthase, okay, I see what you're saying. So could it just be 4 unless there's a debranching enzyme?
No, the synthase is gonna make all this by itself. The synthase, all this addition of glucose, all this addition of glucose to a glycogen, are done by the synthase.
So as long as there's glycogen, it keeps going.
Yeah, it keeps going until it stops about 50,000.
Okay, so all 50,000.
All 50,000, but it cannot do this unless there is the creation of a branch. I'm gonna go there.
that was a trick question.
And you can see here is the whole thing for both the liver and the muscle. You see that? You can see that. And I put the numbers, you can see right here. Look at this. You look at this, hopefully now you understand this better. Because when I look at this, you say, I have no idea what that is.
I'm confused, were we still talking about degradation or?
No, that's
No,
what
synthesis.
I said. This is just to show you that everything is now, since we understand this, hopefully you can understand this now better. The degradation.
Yeah.
That the muscle is converted to pyruvate. And the liver is converted to glucose and release. So it's the combination of the two. But you have everything is there. You can see I'm showing that the debranching enzyme remove the branches. And it's a phosphatase. A hydrolase. And this removes the alpha 160. I want to show you the branching action. So here is the glycogen synthesis. You can see right there, right? What I did was put a metabolite, which I always do that. I put a metabolite, glucose 6-phosphate, right? Glucose 6-phosphate, right? And then we isomerize into glucose 1-phosphate. Why? Because we cannot activate glucose 6-phosphate for this purpose. And then we activate this, and you can see right there. And bingo, here is the glycogen. You can see this is the little bit of glycogen, the primer, whatever it's called. And you can see bingo, what the synthase does is add. But then the debranching enzyme doesn't add more glucoses,
It just continues it.
okay?
Is that every 10?
I'm gonna show you.
Up to 50,000 or?
Yeah, but I think, that was every 10, I don't know. I may be confused.
Here, this is what the synthase does. It makes a very long chain of glucoses, But then the branching enzyme says, okay, I'm gonna take these 7 guys, and with these 7 guys, and then I'll make a branch, and then you continue putting more over here. And then from this branch, you're gonna create 4 by the branching enzyme. That's why this grows exponentially. But the branching enzyme is not adding more glucoses. That's what I asked you the question at the beginning.
It's just rearranging them.
It's a rearranging, making the branches.
But if it's 10%, so is it every 10 glucoses that it does?
Of course.
Okay. I just want to make sure.
On average, that's what it tells us. Okay, that apparently, this is one of the things, the evolution. Yeah, they say that we are branching the glycogen more. I'm gonna show you the structural effect of there is a little table about the glycogen. You see that? Look at this. So I take an average.
Okay.
We discussed this in biochemistry 1, but I know you were never in biochemistry 1, right?
Nope.
That's what I thought.
That was years ago.
That's what I thought.
Yeah. Oh my goodness.
So is it clear now, the
Yeah,
synthesis
it
of
makes
glycogen? And you can see the reactions, for instance. I'm gonna go to the reaction. The reactions are there. But the most important part is that you can take a piece of paper and you can say, I know what happens. I know that glucose is internalized, is phosphorylated, because why do we phosphorylate glucose? Look at this, for this simple reason. This cannot get out. This is impossible. This glucose cannot get out. This glucose is a phosphate. Cannot get out from the cell. Unless we remove the phosphate. So if we want to release this glucose, you basically have to do this, remove the phosphate by the phosphatase. And what is the problem with that? That just a few cells had a phosphatase. Now this glucose can get out. And can get out. Because this is how we release the glucose via GLUT2. Oh, it's the same transporter that we use for internalizing this glucose residue. It's the same one that we use for releasing it. It's the same. but this is a passive transport. So that basically means that when the liver eats the glucose, there is more glucose outside than inside. There is, we are not going to go for a little bit of lasagna. We go for the kill, right? We take, a big portion of lasagna, right? So we put a lot of glucose in the blood. Okay, so this is passive transport. That's what it means, that under fit conditions there is more glucose in the blood than there is, and this transporter is so good that sometimes the concentration of this guy and the concentration is about the same. Because the velocity of transport is so fast and they can internalize. The problem is that the liver cannot store a lot of that glucose. So a lot of this glucose is internalized it's rapidly released and made available to the rest of the body. Okay? It's okay. It's almost like talking to the muscle and to the brain a little bit and to the heart. Hey, I'm sending a lot of glucose your way. Make sure that you convert that into glycogen. Okay? And that's basically what this is. So what I'm showing you is the metabolite. I don't know if you follow my recommendation. Write the metabolite and then you deal with the enzymes. I'm gonna show you. And you can see right there, you can see glucose, glucose-6-phosphate, glucose-1-phosphate, UDP glucose needs to be activated and being the synthase of the branching enzyme. You can see the dynamics of glucose residues doesn't change. Because I'm gonna show you the branching again. Because the branching, the number of glucose residues is the same. Look at this. Look at this. If you count the number of glucose residues in here and in here, it's the same.
Just rearranged.
You see what I mean? However, if you're going to represent, that's why you have to be familiar with the chemical reactions. The chemical reactions, for instance, for the synthase, will look like this. Glucose and number of molecules plus UDP glucose. And the synthase, what it's going to do is basically convert this into glucose plus 1 plus UDP. How many times this is done? I'm gonna say, on average 50,000 times. Because all the glucose molecules are added to the synthase. So why do we need glycogening? Glycogening is needed because often when we prolong the normal fasting between meals, it's very likely that we have degraded all the hepatic glycogen.
So that provides the primer.
And then what? The synthase always need this little thing. You see, the synthase cannot work with this substrate. So, and what the glycogen is going to do is going to make a little bit of glycogen. What is the minimum glycogen, glucoses, that is needed for the synthase to take over? At least 7. And that's what this is what the glycogening is going to do. I'm going to show you. Look at this. Look at this. There is no glycogen, right? So what this enzyme is putting a few glucoses together, as glycogen. But it's not only putting these glucoses, it's almost like I am glycogening, and then basically what it's doing, I'm taking UDP glucose and attaching that glucose to myself. Okay, so I'm making a little bit of glycogen on me, and that's why glycogening is always covalently attached to a glycogen particle, and that's why when we represent a glycogen particle, there is that tennis ball, that's illustration, or there is a G and the other representation. That means, and that's basically, see what it does, attaches this glucose. The glycogening also is going to use You can represent the glycogenic molecule like this too. You see that? But the only thing that you're gonna do that this is glycogen, and then what's gonna happen and this glycogen is gonna have this a little bit of glycogen. This is about, I'm gonna say about 7 glucose molecules. Then once you make this, look at this, because you add this one, right? And then look at this, repeat it 6 more times. So this is the minimum. And there are papers. There was a paper, the person who discovered the laboratory in the UK that discovered this glycogen. The synthase doesn't like it when there is 1 glucose. Doesn't like it when there is 2. Doesn't like it when there is 4. When it has 5, kind of like it. When it has 6, oh, I like it better. When it goes 7, likes it the best. If you add 8, doesn't make any difference. So the minimum for like it the most is at least 7. So that's what is called the primer.
So, say you use the glycogen
Okay?
and everything is done, then that's when we get to the fatty acids and
Wait a
the other
minute.
stuff. Yeah.
I don't know. This it has nothing to do with fatty acid.
Well, no, I was just trying to piece together what happens if you use all the glycogen and the glycogen.
No, you don't use the glycogen in this enzyme. We don't use it. We use the glycogen particle.
So the glycogen, yeah, because the glycogen uses the UDP glucose to make glycogen, right?
Yeah, but to make at least to add about 7 glucose molecules to. So the first, that's what I'm gonna show you. Let me go to the graph and then I can entertain your question.
Okay. I'm just trying to figure out what happens if you do that 7 times.
Look at this. Think about this, which is called the. Look at this. This is the prime, or you can call it first tier. So think about that. This has on average. Look at this. 12 to 14. So half for these guys, if we assume that there are 14 in here, half of these guys were added by glycogen. And then the synthase took over because the synthase can do it faster.
Wow.
Oh.
I get it.
Thank you.
Ding ding
The
ding ding ding.
synthase is gonna do, and then remember that the synthase is gonna do a very long one, but hey, it's time for me to diversify. I'm gonna make a branch. And then it makes another branch from the branch and another branch from the branch. That's why they can make a particle. Cells, liver cells can make a glycogen particle. From no particles at all, you can see the particles after 4 minutes. It's almost like the liver is full with these particles. There are papers about that. Now,
Why didn't you just say so? No, I'm kidding.
Got it. You answered
That's
my question.
awesome.
So the nothing ever happens to the glycogen.
No.
It just uses the UDP to make glycogen in the ground.
glycogen is an enzyme. Nothing happened to the enzyme. The enzymes are like meal vampires that live forever.
Yeah, got it.
You see what I mean?
Ready, I get it.
So,
What is your name?
Haley,
Okay, Haley. Okay, Haley. Stefano.
Angela,
Angela. Freddy. Fassi. Okay.
so then
The
my,
ones who care.
Aww.
but then I guess my next question is probably going in another direction.
Now,
If
go ahead.
glycogen is always present, always able to start this reaction to make glucose residues in that glycogen, then why do we have gluconeogenesis?
Because what happens when this is gone?
But didn't you say it's always there?
No, glycogen in the protein is always there. The enzymes are always there. We might not have the glycogen particle.
Oh, so it needs to bind to that.
For instance, if we during starvation, And you have a conversation with your liver, a serious conversation with your liver. You said, where is my glycogen? He said, I look it up. What do you mean? I send it to your brain. That's why you have. That's
You're
why.
having a conversation.
That's why we're having a conversation. So are you telling me that there is no glycogen left? No. That the glycogen was long gone, some time ago. Okay, so but you are still releasing glucose. Yeah, but that's not coming from glycogen anymore. It's coming from converting lactate and converting alanine into glucose. Oh, really? Oh boy, you are so good to me. When we degrade in the glycogen, we are also engaging gluconeogenesis because both are stimulated by glucagon under normal fasting conditions. So you cannot ask the liver, hey, I'm going to bind glucagon to your livers, but please don't do gluconeogenesis.
So they're not exclusive, it happens.
It happens, I'm gonna show you.
Unless you take metformin, like we.
Shut up, Stefano.
It's true. Like this.
Look at this. This is fasting, right? Look at this. What is this? What is this? This is glycogen degradation. Is gluconeogenesis taking place at the same time? Of course. For instance, just to give you an example, an elite marathon runner, right? They run this marathon in about just 2 hours and a fraction, which is, something is mind-boggling to me. Okay, what's gonna happen? By the time that this guy or this woman goes, I'm gonna say that I run marathons, by the way. So by the go to the mile 20, I'm pretty sure that all the glycogen has gone.
Yep, depleted.
And this guy is still running like an animal. I mean, these guys are still running the mile around the 5 minutes. I cannot run a single mile under 5 minutes. Most of us who can't, they run every single mile, 26.2 miles under 5 minutes each.
That's awesome.
So what is this? Mostly because of the fat oxidation and the little bit of glucose. And most of the glucose that is being released is to keep the sanity of the brain. Because it has a lot of competition by the exercising muscle cells. So, and that's why, for instance, if you are not fully trained, I saw many people running the wrong way in marathons. Because you have to take a break sometimes. And I don't know how these people do it. I remember when I ran a marathon, I had to take up,
Bring glucose tablets.
yeah. No, you have to take a break to urinate.
Yeah.
Because you have to hydrate. But you're gonna be there. For instance, my first marathon, I ran it in 4 hours and 15 minutes. I mean, I never done something more painful, okay?
It's awesome.
That's good though.
You see what I mean? But then I was able to run until my last marathon. I was able to run it under 3 hours, which I only ran 3 full marathons.
Wow.
And for an older guy like me, I ran the marathon.
7.5 minute mile.
It's almost like I ran it in 2 hours, 45 minutes and 15 seconds. I still remember those numbers. Okay. But what is the point? What is the point? The point is that, you cannot ask insulin to stimulate glycogen synthesis without, asking the liver. You're gonna be making glycogen. I'm gonna change this picture. I'm gonna change this picture. I'm gonna put when we have plenty of lasagna. And look at this. What do we do? I fell liver because of lasagna, and here is the lasagna. You see that? Look at this. Look at this. We're making glycogen. We're also making fat. You can ask the liver. Okay, I'm going to buy an insulin to you. Here is the insulin, but please don't mess up with the glycogen or don't mess up with the fat. No, it's going to stimulate both. You see that?
Mm-hmm.
Does it make sense?
Yes, sir.
What else? If you have any specific questions that you still have some difficulties, you know this. But sometimes the point is being able to visualize what's happening. And I think probably you visualize things better now.
Mm-hmm.
Yeah, definitely.
This was a toughie. There was a lot going on. So, from a regulation perspective, the only thing that we have to worry about for this whole entire thing is synthase and phosphorylase.
Well, the synthase, remember that the synthase, look at this, the synthase. The synthase is like this. If we want to make the synthase happen, right? So this is the A, form of the synthase. This is the only one that can actually, the synthesis can go up. But if we somehow put a phosphate on this is gonna be, and now this synthesis that was up, this is not possible. See that?
Yeah.
So you need to know what is the enzyme that convert the A into the B. And there are two enzymes that we discussed here. One is the PKA. Okay, PKA.
PP1.
Okay, and the other one is the glycogen synthase kinase. Any enzyme, in reality, there are like about 20 kinases. See how nice I am to you? Of the 20 kinases I know that can target the synthase, I only show you 2. So, and that's why, and for instance, look at this. Why? Look at this. You know that this is because of fasting. That's why you need to know what are the hitments of fasting, what are the hitments of glucagon. That's why when there is fasting, we bring PKA into the picture. And PKA, you know what it does is put I don't know, the way I show this with my students, it's almost like PKA adds something to you. You know, because what the phosphatase is going to do, which is the enzyme that converts this back into the active form, is PP1. PP1 is a very good friend of the synthase because any phosphatase is going to be a good friend of the synthase because it's removing the phosphate. So by removing the phosphate, it's going to activate that enzyme. You see that? And this is R. That's why this is the immunoinsulin. That's why this is the immunoglucagon, but it's also the immunoepinephrine. You see that? So it's not just the synthase. It's not just the synthase. You need to know that the PP1 is R because of insulin.
Yeah, I know. It was making sense finally why it is.
Oh, why the answer is insulin?
Yeah, I
Yeah.
was referencing your sample test
Uh-huh.
and the question finally made
What time is
sense.
it, guys?
It is 2:45 or 10 to, almost 10 to 3.
Yeah. You see what I mean? So there are graphs that you know that tells you the story. Let me go to regulation. If you understand, to me, when I understood as a student in Venezuela, I went as a guy, if you understand that there is an enzyme that when you put a phosphate, you make it happy. But at the same time that you're putting a phosphate on the synthase, there is a different enzyme that is putting a phosphate on the phosphorylase. Because happens simultaneously, right? See, I'm gonna show you what happened very rapidly. So here is glucagon. So here is the receptor for glucagon. So this is glucagon. Glucagon is gonna bind over here, right, to this 17 receptor. And bingo, I'm gonna put over this. Right away, we're going to make cyclic AMP. Cyclic AMP is gonna activate PKA. PKA, right away. Know what is shown in the book. Right away, it's gonna start phosphorylating because that's the only thing that this enzyme can do. And right away, it's gonna phosphorylate the synthase, right? So the synthase is going to be converted into synthase with a phosphate. Bingo is gonna kill it right away. This guarantees that there is no possibility of glycogen synthesis because this is the enzyme responsible for synthesis. So right away, this synthesis is inhibited, right? But then PKA is going to make phosphorylase kinase. I'm gonna put 3 letters, glycogen phosphorylase kinase happy. And this is the enzyme that's gonna make the glycogen phosphorylase. It's gonna have a phosphate, bingo. This is gonna be up now. That's why the same hormonal pathway that kills the synthase is the same one that activates phosphorylase. And now what's gonna happen? That now this, degradation is going to be up.
Makes sense.
This is the key. You have to do this.
What also makes sense with the enzymes, 'cause
No, what I'm saying is, what I'm showing when you regulation, you have to write it on your own ways of writing things. You cannot study these reactions or this process isolated from each other. This is happening simultaneously. And this is not the only 2 target for PKA I'm gonna show you. This is also targeting the bifunctional enzyme of glycolysis. So that basically means that the bifunctional enzyme, that basically means that fructose 2,6-bisphosphate is gonna go to 0. Okay, and I'm gonna show you that it's also gonna target fat mobilization. I already showed you that. But since we are limiting just to the glycogen aspect, I'm showing you the. So the point is that you have to do this anyway, but make sure that you see the logic. If you don't see the logic, something is missing.
Can you generalize and say that any kinase that would attach a phosphate to a synthase would inhibit the synthesis?
No.
No? Okay. Okay.
Some enzymes are activated by phosphorylation, other enzymes are, and that's basically, but they might be a little bit of true on what you say. Now, I'm gonna switch gears, right? But rapidly, I'm gonna show you why when we eat lasagna, there is no possibility of degradation. So here it is, right? So here is the lasagna, right? And that's gonna bring insulin up. So here is the receptor for insulin, right? Bingo is gonna bind with. What's gonna happen? What is the heme for insulin? PP1. See? So what is PP1 is gonna do? I'm gonna show you what PP1 is gonna do. It's gonna target 3 enzymes of glycogen. Metabolism, right? It's gonna target the glycogen phosphorylase kinase. The glycogen phosphorylase is gonna be is down. You see that? the glycogen phosphorylase is gonna be with no phosphate, and it's gonna take down. What's gonna happen? What is this? These are the key enzyme for degradation. So glycogen degradation is gonna be inhibited. And then what happened? Now there is an enzyme that's very, very happy, and that's the syntax. And bingo, what's gonna happen? Synthesis is gonna be stimulated. It's happening simultaneously. You cannot ask PP1, no, leave the glycogen phosphorylase alone, leave the glycogen phosphorylase alone. No, it's gonna target all these enzymes. Oh, I was looking for you. I said that phosphorylase use a machete to remove the head. And then, showing that, that they were removing the head is removing the phosphate. There is a movie, a really bad actor, Mexican, I like him, so we play bad guys, and that's the guy in that movie. Okay,
Thank you.
does it make sense of this?
Yes.
Better than what we got here.
And remember, you can always email me if you're dealing with